with this concept of a CAM cycle. And so we were talking about this last time, how these CAM plants are dealing with extreme hot conditions during the day, thinking about the desert environment, very low water availability, certainly not on a regular basis. And so they have done a pretty extreme shift on how they do photosynthesis in the sense of this day-night rhythm. And so you're seeing it, during the dark. It makes sense that if you're living in a desert, don't open your stomata during the day because you might dehydrate and die from that. And so take in your CO2 at night when it's cooler, a little bit more humid, but they can't do photosynthesis during the night. So they have to just stuff all that away into the vacuole. And so they run the first part of it where they take in the CO2 and with the help of that pepcase enzyme, they then react the CO2 and the phosphoenolpyruvate together, and that gives you oxaloacetate, which gets converted to malate, and then that gets stored in the vacuole. So carbon dioxide is part of that malate molecule. And then during the day, go ahead and shut down your stomata because you're going to lose too much water. And now that the sun is striking your body, you can go ahead and get that malate out of the vacuole, decarboxylate it, so cleave off the CO2 and feed that to Rubisco. So now that enzyme that is kind of sloppy, and it's seeing lots and lots of CO2 and very little oxygen. So it will do the appropriate thing of running the C3 cycle or the Calvin cycle if it keeps getting fed all that lovely CO2. And then the other product of the decarboxylation of the malate is pyruvate. And so in the case of CAM plants, they will then just stuff that into starch. They'll make starch out of that and just keep that building up in the chloroplast all day long. And then at night, they'll go ahead and start breaking down the starch to provide the phosphoenolpyruvate. So we've been looking at that. Are there questions on this pathway or cycle? Okay, so when you look at it and say, why would a plant do this? Well, first of all, it's huge conservation of water. And then the other payoff similar to the C4 cycle is that you're concentrating the CO2 around Rubisco. You're making sure that Rubisco is seeing plenty of carbon dioxide, very little oxygen. And so under those conditions, it will run the Calvin cycle, which is good because that's how they get their sugars made. And so I think we left off. Okay, so we covered this idea how some CAM plants can be, facultative, which means they could take it or leave it. So under good water supply, they might decide to not run the CAM cycle. Under normal desert conditions where you have lots of drought and lots of heat, then they would go ahead and run. And so that would be like pretty sweet if you could turn it off and on, because there is an extra, energy expenditure to run the CAM, but it's definitely worth it for those plants that are adapted to the dry conditions of the desert, dry, hot conditions. And so I think I mentioned already it was named after the family Crassulaceae, and so there's 3 different genera as examples within that family that can do the CAM cycle, but there's many others, about 38 different families, and it is not unique. So, one angiosperm family can do all versions of how they make their sugars, whether it's all plants do C3, but then you can have those that can do CAM and those that can do C4. About 60 different times through the points of evolution, how this CAM cycle has been manufactured. And so you have lots of individual opportunities through the history of evolution where this has been showing up. Some really important plants with regard to commercial production, but they also can find this showing up in more primitive plants. And when I say more primitive, I'm thinking the seedless vascular plants. No, the Crassulaceae family, know that name. But I'm just giving some examples, but I'm not gonna ask you to like spit those back to that. That's okay. And so, and just knowing that lots of different families can do this and that it's have evolved many different times through history, I think those are important concepts to think about when you look at C3, CAM, C4, that's pretty much the order that they see how it evolved over time. You know, every plant can do C3, and then CAM came along that pathway, and then C4 came last of the three. And so this is taken from a paper that was published a few years ago, and pretty much saying what we've been discussing about the different families of plants that can do this, and that um, you have this idea of really dry, hot conditions driving this adaptation. That concentrates the carbon around the rubisco. But even aquatic plants, it sounds kind of weird, but there's some, like, so one of the, see the vascular plants that runs the CAM is called Isoetes. And we did talk about that during the vascular plant section of the class, and it's aquatic. So it's kind of weird. You're like, really, CAM? But it's aquatic. It's not under any drought conditions. But CO2 as a gas does not dissolve and diffuse very readily in water. So water in general, when you think about CO2 and oxygen dissolved in that, it takes a while to diffuse through. And so they think that even though it's an aquatic plant, it still struggles with access to adequate CO2. And so it did develop the CAM pathway. So this slide is kind of putting it into context of the evolutionary process. And then this is one of the pictures from that paper, just trying to think about periods of time through evolutionary history where CAM, the evolution of the CAM pathway was probably, you know, super relevant. And so thinking about any kind of desertification, I think BP is before present time. M stands for millions of years. And so then you have times in our evolutionary past where CO2 levels are dropping. So again, it's scarce. And then you think about all the plants that we've cultivated over the last 12,000 years. And so it does allow for all of these conditions, allow for plants to adapt by creating this, you know, the CAM pathway to deal with drought and low CO2 concentrations. And then this final paragraph that I put in is from the paper as well. And pretty much saying how, you know, we need to understand this better, especially with climate change happening and the idea of more and more agricultural areas might become consistently warmer and drier. And so again, there's this interest in, if we can better understand the CAM pathway, can we engineer important crop plants to perhaps have this adaptation through synthetic biology and genetic engineering. So they're looking at that. And then this is the front abstract of the paper. But basically just saying that, you know, we need to look more at CAM plants more carefully and see what we can do about that in terms of, can we use that knowledge to create plants that might be better suited to conditions we're creating with climate change. Really good question. I don't know the answer to that. I would think it would be a little bit easier actually to do the CAM C4, but in terms of the physiological, however, that day-night rhythm, that's a big switch. So I don't know. I mean, I think they're further along. I'm not quite sure why they decided to hone in on C4, like the C4 rice project. Yeah, right. Yes. So that's another big thing with C4. It's like leaf anatomy is a big deal. With CAM, when you look at what's going on in the cell, you know, the whole pathway happens in a cell. you don't have that difference in spatial, but that difference in time of when things happen. So yeah, I don't know. I don't in that, and I looked, maybe I didn't use the right search term, but I couldn't find any groups working on like engineering for CAM. I found it for C4, but not CAM. So I would think someone's doing it by now, but maybe I just didn't stumble on it, the right search term. And I think I showed you this already with the difference in water loss between the 3 cycles. And so you can see C3 is the least efficient in terms of conserving water. And CAM is definitely the best at conserving water. And then, okay, so we've looked at the C3 cycle. We've looked at when things go wrong in the PCO cycle. Then we looked at how plants have adapted with the C4 and CAM. But all together, let's kind of finish the thought. So they're doing all of this, and they're doing that to make their sugars. All right, that's their goal through all of this. Is let's make our sugars or create starch. And so if they have plenty of sugars ready to go and they got plenty of sugars being translocated through the phloem, then they might start taking a portion of those sugars and converting it to starch, or kind of saving up for a rainy day in case things get desperate. They can have that stored starch as a source of carbohydrate when needed. If they choose to do that, it's going to be in the chloroplast. That's where they stuff starch. It gets made in the chloroplast. It stays in the chloroplast for storage. A few plants will decide, so to speak, to store their sucrose in the vacuole. They won't convert it to starch. They'll just store it as sucrose. So there's a couple of plants that do that are important. Oats, wheat, and barley in terms of crop production will do it that way. The plant may also say, you know what, I need some more sucrose. And so if they choose to do that, then they have to get their initial sugars that they're building through the C3 cycle. All that's happening in the chloroplast. So then they have to get it out to the cytoplasm and then run a pathway for sucrose synthesis. Then, of course, once they get their sucrose, a big job is to get it exported throughout the whole plant to areas that are sink tissue, from source to sink. And so moving through the phloem, getting it from source to sink, and then all parts of the plant body will use that sucrose, whether it's source tissue or sink tissue. They're going to use that sucrose for metabolism in order to get their energy needs met. They're going to use that sugar for maintaining their cell structure. They might need to build certain components that need those sugars, or they might store up. So once the sink tissue gets that sucrose, they might decide, you know what, I'm going to store it and have some stored up in case we run into deficiencies. So the goal is to get, whether it's C3 CAM or C4, it's to get either starch or sucrose built up and translocated throughout the plant body. This is just trying to show in one cell, when one of the leaf cells is doing photosynthesis and they're doing the Calvin cycle, they're showing how that's happening in the chloroplast up top here. So there's the Calvin cycle. And then you have your the triose phosphate, that's essentially your G3P of the Calvin cycle. And so that can get put out into the cytoplasm and it can be used for sucrose synthesis. Or that G3P can be used to run the pathway in the chloroplast for starch. But in one cell, it's an either or idea. Okay, it's not going to run both at the same time because it's controlled very tightly. One of the ways it controls it is by this transport protein in the membrane, and you have this phosphate coming in as the G3P is coming out, and so it's like this one-to-one transfer through that protein. And so this is pretty tightly controlled in terms of, should I make sucrose? Should I do starch? And then lots of controls also on this sucrose synthesis pathway that I'm not going to get into. And so I don't expect you to know the details of sucrose or starch synthesis, just know where it happens. And the idea that it's kind of this either or within one cell. It's not being done at the same time, so to speak. It's an either or relationship here. So either or, one or the other. You can't do both at the same time. All right, so basically this triose phosphate, what they're talking about is that all this G3P, where they show this one going for export, that's what they're talking about, that you get the G3P exported out to the cytoplasm for sucrose. So that's what they're talking about there. Okay, so we've covered all of the ideas of photosynthesis that I can get into in this class, both light reactions and then the biochemical reactions to make sucrose. Any lingering questions on that right now anyways?
One of the things
Mhm.
that can be sort of what makes
Yeah, that's a really good question. The oat sweetened barley, like why would they just stuff it into their vat? Well, you'd think, I mean, they must do some level of starch synthesis too, because that's a really good way to store up extra sugar for long-term storage. But I couldn't find an answer. I've been wondering that too. Why would they do that strategy? And then, you know, like they often mention these three because they're important to us commercially. But it begs the question, what other plants might be doing this? Is this like really common or is this really unusual? And so if I can track down that information, I'll give an update. But
but do they have the start like
in the core class?
Yeah.
Yeah, I think I find it hard to believe that they wouldn't do some level of starch. And if they do starch synthesis, it should be happening in the chloroplast because that's where all the enzymes are located for those. But maybe they just delay starch. Like maybe they don't do that as frequently as most other plants do. Like they kind of hedge their bets and I'm just going to keep it as sucrose and stick it, stick it in my vacuole because I might need it right away, type thing. Whereas starch is more like long-term solution, like socking it away in a bank account. I'm not going to touch it for a while. So, any other questions? Okay, well, we're going to shift gears a little bit now because the next topic I want to cover is on mineral nutrition. So we're still thinking about how plants get their nutritional needs met. With photosynthesis, it's their carbohydrate needs. But now we need to look at, you can't survive just on carbs alone. You have to have minerals as well. And so the next slide set that I just posted that right before class, is on mineral nutrition. It's in EDUCAT. And so I know that you're running that mineral nutrition lab exercise looking at deficiencies, but we need to think more like big picture idea of what's going on with mineral nutrition. By the way, this is a mural on the side of a co-op I think in Ashland, Wisconsin. And this is just a portion of it. It's a huge wall across the whole side of the building. And I got so excited because they actually focused on stuff going on underground. Usually when you see paintings, it's all above ground stuff. But you can see all the brown here. They're trying to depict what might be happening in the soil environment, including mushrooms. I loved it that they had all the mushrooms drawn on there too. So I thought that was really cool. So that's what we're going to do. We're going to dip into the soil and we're going to think about how a typical plant who has its roots buried in the soil, we've already talked about this idea of transpiration and that we have water moving through the plant, but don't forget it's water plus the dissolved minerals moving through the plant. So now we're going to zoom in on the dissolved minerals part and think about, okay, when the plant is going through transpiration, one of the huge benefits is that they're bringing in these dissolved minerals. and getting that into their plant body, and of course, nourishing the different cells and tissues of their body. So we're going to zoom into the soil again and think about the dissolved minerals in soil in general. And so many of you I know have had the soils class. And so when you start thinking about soil, it is extremely varied in composition. But let's say some basic things about it. So you can have the solid part is, of course, inorganic particles. And basically those particles that we see in the soil are a reservoir of nutrients, of minerals that the plant needs. And so, potassium, calcium, magnesium, and on and on and on. We also have organic particles in soil too that are coming from living organisms that are sloughing off cells. Remember, roots are really kind of leaky. They exude chemicals. They slough off root cells. In particular, you have dead and dying animals and organisms. So all of that is contributing to the organic particles. And of course, when you look at organic, living creatures that have died and are starting to decompose and get recycled, there's a lot of nitrogen, phosphorus, sulfur in those organisms, whether it be plant or animal or whatever. Then you have the liquid phase of the soil, and that's what we're going to zoom in on for how the plant can get its needs met, because plants have to have those minerals dissolved in water before they can get access to it. They can't just knock minerals off the soil particles. They can't do that for the most part. It's just, they can't do it. And then don't forget, there's gases that are dissolved in the water of the soil. Certainly, pretty much all the gases that we see above ground are below ground as well. And then there's other organisms in the soil. There's creepy crawly insects and organisms. There's the bacteria and fungi that are all doing their gas exchange. They, oxygen, CO2, there's hydrogen sulfide gas. There's lots of other methane. So I've only listed the big ones, but microbes can also contribute some other gases in particular to this list. And so we got all that going on. Soil is extremely complex and varied. And, when you think about what the plants have to do, it's quite amazing that they can get their needs met through this process. But basically what we're looking at is when you look at soil particles, no matter what soil particle you're looking at, basically all those studies have shown that soil particles are negatively charged. They have an overall negative charge. And so this picture in the middle here is showing how you have these different minerals attached to the soil, but you can see all these minerals are positively charged. And that makes sense if you understand, basic laws of chemistry, that opposite charges attract each other, right? So the negatively charged soil particles will bond with positive mineral ions or cations. So here you're seeing some examples of that in that middle picture. So the negative charges, basically it's coming from oxygen as part of the carbohydrates that are in the soil. It's coming from oxygen that is part of protein and carboxyl groups and phenols. So all of this adds up to overall, you have a negative charge on these soil particles. And then depending on the soil, you look at the surface area to volume ratio. And, there's a lot of soil particles that have a really decent surface area to volume, which is good for the plant, because then that means there's a lot of cations stuck to the soil particle that the plant can eventually get access to. And then that table over there on the right is just looking at the mineral elements that the plant will be using. And you can see on that list that there are plenty of cations, the positively charged, but you see a few on there that are negatively charged. So plants need both. They need cations and anions for their mineral nutrition. And they have to have different strategies in order to deal with that idea. But basically, if they want to get access to any of these minerals, whether they're positive or negative, they have to be dissolved in the soil water. They can't just like somehow knock calcium, for example, off of the soil particle all by themselves. They just can't do that. And so, but once it does get dissolved in the soil water, then the plant root can take it up. So then that begs the question, well, then how do these minerals get into the soil water? If the roots aren't actively somehow, grabbing off the minerals from the soil particle, how are they getting dissolved into the water? And basically, we're thinking about two big ideas. Actually, one, this is usually the one that gets mentioned, which is accurate. Cation exchange process is a method that we'll see. I'll tell you about the basics of it. It's a way for the cation to get bumped off of the soil particles and get put into the water. So it's called the cation exchange process, and that's absolutely accurate. However, a lot of people forget this second one, microbes. All right, microbes in a healthy soil, you have plenty of microbes that are really capable of knocking and grabbing these cations off the soil particle. And as they're doing that, a lot of it will go into solution, into the water, dissolved, and then the plant can get access to it. So when you look at mineral nutrition for the plant, it's a team effort. There are plenty of microbes in the soil that are helping with this process. And often this gets overlooked in a lot of different textbook readings or articles that I've read. So my big push is for you to understand that plants are certainly not living in isolation when they carry out their lives, and that they have all kinds of microbes in the soil. They have all kinds of microbes in and on them that are going to be helping them. In this particular case, we're thinking about the soil community and how microbes are in the soil by the roots, helping with this mineral nutrition idea. So that plate that you're seeing on the picture there, that is a soil dilution. I took a soil core from around some plant roots and diluted it, and then I spread it out across an agar plate and let it incubate and grow. So all those different colors are different species of microbes. There's some bacteria and fungi on here. Lots of different pretty colors. All right, so just to show that soil, and this is just one basic agar recipe. This is showing a tiny fraction of what's really in that soil that I took. I weighed out 1 gram. And so there's millions of microbes in 1 gram of soil. And then, we also know that there's microbes in and on the plant. And so this lower left picture is looking at a leaf section that's put onto tissue culture medium and those fungal endophytes coming out of the leaf. And so microbes, I found this quote very fun to think about in this paper. They say root systems, which absorb nutrients and are directly exposed to the soil microbes, are functionally analogous to the animal gut. I really like that idea. And I think it's accurate. It's a useful way to think about what's happening in the root zone and the roots with its associated microbes, because there's all kinds of microbes that have attached to the root surface. There's all kinds of microbes that are just like a couple of millimeters away in the soil, right next to the root. The population densities of microbes in the root zone is tremendous compared to what you see in just normal soil without any roots. Huge difference. And so the plant does cultivate its own collection of microbes in its root zone, and those microbes are really, really important for the mineral nutrition aspect for the plant. Okay, so don't forget about the microbes, but with cation exchange, we need to think a little bit about that. So cation exchange, what they're saying is that when you look at a soil particle, you can have cations coming off of it and other cations replacing those that are moving off of it. So it's exchanging. And so here you're seeing a picture where you have a clay soil particle. You see all these cations attached. And now if you can imagine like rain coming through the soil system where water's percolating through the soil, you have some dissolved minerals in that water solution and it's percolating through. And you can imagine where some of those dissolved minerals will make contact with this clay and they can actually bump off some of these cations and replace them. And so here you're seeing a couple of ideas. They're trying to show, like in this case, they're showing H plus, a proton, is coming on to the soil particle and bumping off a potassium. Okay, so that is cation exchange. That proton has exchanged onto the clay, and potassium is coming off of it. So that can happen with just water movement through the soil with rain events. You have, dissolved minerals in the soil just coming into contact with all the soil particles and doing this exchange process with the cations. The other issue that you're seeing on this image is that when the plant roots, remember this is living tissue, so they're going through respiration, similar to what we do. They need oxygen. The plants need oxygen for their respiration. And then you see that they do give off CO2 as part of their respiration process. One thing to be aware of is that when you mix CO2 and water together, it creates this reaction that they're showing. And one of the big products here is H plus, or protons. Protons, remember now, when you look at the pH of any system, the pH is dependent on the concentration of how many protons are in that system. And so we'll be thinking about that a little bit too. So keep that in mind, that there's plenty of protons that are getting made just through the roots, going through respiration, and when CO2 gets released, you get this reaction happening over and over and over in the soil environment. And it can certainly contribute to cation exchange. So they have these so-called lyotropic series that they have put together. Basically, it's just a way to describe which cations are capable of bumping other cations off. And so this is an example of a lyotropic series. So the way you interpret these series is that what you find on the far left has really high binding affinity for soil particles. And then as you work your way off to the right, the binding affinity gets weaker and weaker. Okay, so what this means, so that's what the greater signs are supposed to be. Aluminum is greater than the proton, which is greater than calcium, which is greater than magnesium, and so on. So what this means is if aluminum is dissolved in the water and it's flowing past this clay particle, it can easily bump off anything to the right. So it can bump off protons, calcium, magnesium, potassium, ammonium, sodium, all of those. It can bump all of those off because it has such a good binding affinity for the clay, for the negative charge. And then like calcium can bump off magnesium, all the stuff to the right, but it can't really easily bump off the proton or the aluminum. Usually it's a 1-to-1, I think, but yeah, I don't know. That would be a question for Dr. VG on that one, because with the 3+, my assumption, the way I've always seen it described, is it's 1-to-1, but I don't know. So again, it's just, you can look these up. They're, they figured out which ones have higher binding affinity, which ones are lower. And so, but you can see in this, in most lyotropic series, protons are pretty good at bumping off lots of other molecules, or minerals, I should say. Okay. So, if you've taken soils class, you know that there's many different types of soils, different ratios of the different soil particles and types of soil particles. This is just looking at a few different clay types. And so here they're looking at morillonite, ileite, and kaolinite, and just looking at comparing those 3 types of clay. And on the far left column, you're seeing, by size, shape, their cohesion properties. And then the very last on the row there, on the column there, is cation exchange capacity. And so you can, like, if you're growing plants in your garden, you can take a soil sample and send it off, and you can have them do these types of analyses on your soil. You can find the composition of your soil. Do you have heavy clay, heavy sand? Are you more in the loam arena of soil mixtures? And so they can give you all of that. And then one of the other really nice pieces of data that they can give you back is the cation exchange capacity, because you want a really decent cation exchange capacity if you want to grow plants. And so here you can see with the morillonite, you have really decent numbers of your cation exchange, but then you see a big difference as you look at the other two. And so growing plants, you want plants to have a system, a soil system that has really decent cation exchange so that they have a constant and kind of a steady and varied supply of minerals that they can get access to. All right. And then you can see too with the different sizes of the particles that definitely influences all these other properties that you see in the table here. So, and that's just looking at clay. There's all kind, there's sand, silt, clay, and then you have to factor in the humus and organic matter, and it's complicated. But basically, we're thinking about this idea of with cation exchange. If you have that soil system, if it has a nice cation exchange, that means your plants are probably getting well fed in terms of a varied diet of minerals and a steady diet of minerals. Right? So if you ever do this and get a soil analysis report back, pay attention to that. And there's ways to improve it. So if you find out your soil system's poor in cation exchange, you can add in the right types of soil particles in the right ratios for sure. So it's not like you're stuck with that. But here again, you're just seeing this idea of cation exchange with the soil particles. All right, so cation exchange means you have the positively charged, but we know plants need negatively charged minerals. So that's something else to think about. So if they do need anions, but anions aren't going to attach to the soil particles. So that's the problem, potentially. On the one hand, if they don't attach to the soil, that means they're always available in liquid water, right? So, I mean, that's good. On the other hand, if the plant roots, so if you imagine water percolating to the soil, whether it's a rain event or irrigation of a commercial field, eventually that water is going to leave the root zone. And if the plant didn't take up those anions as the rain is passing through their root zone, then all the anions keep going. There's no like reservoir or bank of anions in the soil. And that's a problem because plants, especially nitrogen, unfortunately, nitrogen is one of the big ones that's an anion, and nitrogen is huge for the plant. So yeah, it's, on the one hand, that it stays in solution, that means the plant has ready access to it. So that's good. But again, if they don't get that captured and brought into their tissues well enough, quickly enough, all those anions can percolate through down into the water table. And so, for example, nitrate, NO3 minus, that's often the form that plants prefer for nitrogen. They take up nitrate from the soil, but it's negatively charged. So if you add fertilizer, any kind of nitrate fertilizer, you better know what you're doing, because if you overdo it and now that nitrate doesn't get taken up by the roots and then it keeps going, you're polluting because you're putting all that runoff fertilizer into the water table, the rivers, the lakes, and that can cause a big problem. Big fancy word, eutrophication. You probably heard about that at one point or another. And so this eutrophication idea is a big problem. And now with better technology and better understanding of this problem, things have gotten better. Like the precision fertilization schemes that some of the bigger farms can do because they can afford the bigger the fancier technology, where they can monitor nitrogen levels in the soil quite readily in the soil water. So it's getting better for sure, but we got a ways to go. So anions, it's a different situation. So they're trying to show, this picture is trying to show that you have the anions just kind of out in the water solution. They're never attached to the soil particle. Okay. All right. So let's go back to those protons that can be found anytime the plant is going through respiration and producing CO2. When that mixes with the soil water, one of the products are protons. And so remember that the soil pH, or pH in general, is dependent on the concentration of protons, the negative log of that. When they look at all the different soil systems, the general rule of thumb is that it seems most minerals are available in this pH range, 5.5 to 6.5. And most commercially important plants seem to prefer that. There are definitely some exceptions, but this is a good rule of thumb. So I'd like you to know that pH range and commit that to memory. So pH 5.5 and 6.5, most plants prefer that. And so remember, again, it's the pH is the actual definition of that. It's the negative log of the proton concentration. Okay. So what they mean by why do plants like this 5.5 to 6.5, it's because the minerals are more available in that pH range. The cation exchange process is cooking along really nicely in that range. A lot of the microbes are happy in that pH range as well. And so you get them thriving and doing their business too. And one thing to be aware of, though, that's kind of special is that fungi really are well adapted to acidic conditions. So, 5.5 to 6.5, that's slightly acidic. And fungi are well adapted to that. And that can be good or that can be bad because there's plenty of beneficial fungi that hang out with plants, as you know, but there's also plenty of pathogens that can do some definite damage to plants. So hold that thought. But first, before we get into that, so here on the right, you're seeing the different minerals. And the way to interpret this, so on the x-axis here is the pH range. And if you go and look at the range 5.5 to 6.5, if you look at that and then kind of work your way up through the, let's see if I can do this without, I've had a lot of coffee today, so maybe not, but let's see. Oh, not too bad. So in that range, you can see how fat every line is. The fatter the line means it's more available. All right. So manganese really drops off in availability. Where is that? Between 7 and 7.5. That's what that means, where it tapers down like that. That means it's not very available. Okay. But the 5.5 to 6.5 range, you can see most minerals are really nicely available. Okay. So we're looking at these ideas and then thinking about the cation exchange process and then the ability of the microbes to do their thing. I just want to bring back the idea of don't forget about the mycorrhizal fungi in the soil system. We've already talked about fungi and mycorrhizal fungi, but I need to bring it back again for this concept because the mycorrhizal fungi are hugely important in helping the plant get access to key minerals. And this, I love this picture because it's nicely showing the size difference between the roots in with the arrow here, the root system versus the fungal hyphae and mycelium that's growing off the root into the soil. And if you can imagine it scarfing up the water and dissolved minerals, fungi are also very good at solubilizing. Ooh, that's a hard one. Solubilizing minerals. So that what that means is taking the minerals off the soil particle and getting them in the solution. So fungi and bacteria are pretty darn good at that. And so what I thought was amazing. This paper was published a couple of years ago. I don't think I showed this to you yet, did I? Where they're finding mycorrhizal fungi. So these are arbuscular mycorrhizal fungi. And they know that mycorrhizal fungi are really good at getting access to phosphorus for the plant. But now they're realizing that it's a team effort. Mycorrhizal fungi have bacteria moving along their hyphae, and they go out and hunt down the phosphorus in the water. This particular paper was looking at how the fungi and its associated bacteria are helping solubilize the phosphorus, and then the plant can get access to that. Let me see if I can play this really quick. So this is a picture of the YouTube video where you can see the hyphae, and then I don't know if you can see all the little rod-shaped things here that are right on the surface of the hyphae. Those are the bacteria that are kind of using the fungi as like a highway system. Let me show you a picture or the video. It's short and it's really cool. So they're finding that they move through the soil as a team effort. There's a film of liquid water on the hyphae that the bacteria are essentially skating in or swimming across the hyphae. And so it's allowing the bacteria to have really rapid movement as the mycelium are growing out into the soil. And it's both the fungus and the bacteria that are solubilizing the phosphate, so the plant can get access to it. And so this was just published a couple of years ago. So once again, you know, the idea of you can never get bored with this kind of stuff. There's always some bizarre new thing to think about in some new discovery. Isn't that cool? So you have this team effort where, so again, don't forget about the microbes. Yes, cation exchange capacity is huge. However, microbes are also doing a lot toward the idea of helping the plant get access to these very important minerals. Okay, we got to stop there. We'll continue on with this idea on Friday.